Welcome to Microbiome Signatures, where we examine the mechanistic underpinnings of chronic disease through the lens of the microbiome. I'm Karen Pendergrass, a microbiome signatures researcher, and today we're discussing a critical environmental determinant of neurodegenerative disease, heavy metal exposure, and its implications for late-onset Alzheimer's disease, load. While Alzheimer's is often framed within a genetic paradigm, particularly with the ApoE4 allele, an increasing body of literature suggests that environmental toxicants, including heavy metals, play a crucial role in disease pathogenesis, likely through oxidative stress, neuroinflammation, and microbiome-mediated detoxification pathways. The intersection of metallotoxicology, the gut-brain axis, and neurodegeneration is still an evolving field, but emerging data suggest that microbial composition modulates both susceptibility to heavy metal burden and subsequent cognitive outcomes. So let's break down the mechanisms at play. Let's talk about heavy metals as drivers of neurodegeneration. From a neuropathological perspective, Alzheimer's disease is characterized by the accumulation of extracellular beta-amyloid plaques and intracellular tau tangles, accompanied by progressive neuroinflammation and neuronal loss. Heavy metals, particularly lead, mercury, arsenic, cadmium, and aluminum, are potent disruptors of these processes. Next, let's talk about metal-induced oxidative stress and neuroinflammation. Heavy metals are known to act as pro-oxidants, generating reactive oxygen species ROS and reactive nitrogen species RNS that impair mitochondrial function and trigger lipid peroxidation within neuronal membranes. Lead interferes with calcium homeostasis, leading to excitotoxicity via dysregulated NMDA receptor activity. And mercury directly induces neuroinflammation by activating microglia and upregulating pro-inflammatory cytokines, such as IL, TNF, and IFN. Arsenic crosses the blood-brain barrier and disrupts neurotrophic signaling, impairing BDNF brain-derived neurotrophic factor expression, which is critical for synaptic plasticity. Now that is covered, on to how heavy metal oxidative stress and neuroinflammation lead to downstream concerns with protein homeostasis. Let's dive in. All right, so heavy metals interfere with protein folding and clearance mechanisms, accelerating beta-amyloid aggregation and tau hyperphosphorylation. For example, aluminum binds to beta-amyloid and enhances its fibrillogenesis, increasing plaque deposition. Cadmium inhibits proteasomal degradation, leading to an accumulation of hyperphosphorylated tau and increased neuronal apoptosis. Cumulatively, these disruptions establish a neurotoxic milieu that primes the brain for accelerated neurodegeneration. As you can imagine, that's a problem. Now let's get into my favorite part, which is the role of the gut microbiome in metal detoxification. A key but often overlooked component in heavy metal metabolism is the gut microbiome. The intestinal microbiota plays a dual role in modulating metal toxicity, either by facilitating excretion or exacerbating systemic bioavailability. What most folks don't know is that microbes play a central role in the biotransformation of heavy metals. In other words, certain gut microbiota taxa possess metal binding properties, effectively reducing systemic absorption. For example, lactobacillus and bifidobacterium demonstrate high-affinity metal chelation, particularly for lead and cadmium. Desulfovibrio, I really hate saying this taxa out loud, so apologies if I butchered it. Desulfovibrio are sulfate-reducing bacteria capable of precipitating mercury as insoluble HGS, reducing systemic toxicity. Now, conversely, gut dysbiosis may actually increase heavy metal burden, as a dysbiotic microbiome is often deficient in these protective species. So that makes sense, right? Then there's the heavy metal-induced dysbiosis and gut permeability issue. Heavy metals disrupt microbial equilibrium, favoring pro-inflammatory, pathobiont-enriched states. So for example, lead exposure reduces bacteroidetes while increasing proteobacteria, a shift correlated with increased gut permeability and systemic inflammation. Cadmium toxicity enhances lipopolysaccharide, also known as LPS production via increased Enterobacteriaceae, further aggravating neuroinflammatory cascades via the gut-brain axis. Altogether, this suggests that an altered gut microbiome signature may not only serve as a biomarker for chronic metal toxicity, but could also mediate susceptibility to neurodegenerative progression. But this is all essentially primer, because the important part are the clinical implications. Well, there are major clinical implications here. For one, understanding the role in metal metabolism has major clinical implications for Alzheimer's disease in terms of prevention and intervention strategies. And for two, understanding the relationship between those heavy metals and how they support pathogenic species that are involved in the pathophysiology and progression of neurodegenerative diseases is also vital. So while we assess patients in clinical settings looking for genetic markers or family history, perhaps it would be helpful to ask, where did these patients grow up? Or where do they work? And perhaps even what their diets look like, because, that can be such a huge environmental factor, right? The diet, smoking, and we should most definitely be discussing strategies for reducing heavy metal exposures. Luckily, there are microbiome-targeted interventions like chelation therapies, prebiotic interventions to increase certain species like Lactobacillus rhamnosus and Bifidobacterium breve, which demonstrate protective effects against lead and cadmium bioaccumulation. There's also stuff like clinoptilolite zeolite, which is an aluminosilicate powder that you can basically put in your food and drinks that has demonstrated efficacy in binding lead, cadmium, and arsenic, which potentially reduces the neurotoxic impact of foods. But the point is, given the substantial role of heavy metals in Alzheimer's pathology, future clinical interventions may increasingly incorporate microbiome-targeted strategies alongside traditional therapies, which I am so here for. So for closing thoughts, I want to say that the relationship between heavy metal exposure, the gut microbiome, and neurodegenerative disease is complex but increasingly supported by mechanistic and epidemiological evidence. If we aim to mitigate late-onset Alzheimer's risk, addressing environmental toxicant exposure and microbial resilience should be central to our approach. But, and I cannot stress this enough, we are all going in blind if we aren't spending enough time trying to understand what the microbiome signatures of these neurodegenerative conditions are trying to tell us about the environment, the pathogens, and the relationship of the pathogens. Like, what do they have in common? A lot of times we will find that they all thrive in conditions with these heavy metals or have advanced metal acquisition systems or detoxification mechanisms, or simply that they are used as cofactors for virulence factors. But as always, I just want to encourage researchers and clinicians to stay informed as this field evolves. For more microbiome-related insights, please visit Microbiome Signatures and subscribe to our research updates. We are trying to close this ridiculous information gap and get clinicians and patients some real practical microbiome-targeted interventions for these god-awful conditions. So until next time, keep investigating the intersections between the microbiome and chronic disease. And just remember, the future of medicine is not what it used to be. Thanks, everyone.